So I will start. I would like to present the research conducted in cooperation between a scientific institution and an industrial partner. The scientific institution is the Institute of Biochemistry and Biophysics of Polish Academy of Sciences. Next slide, please. And the industrial partner is the Polish food company that operates in various branches, including the sugar industry. The company has got 7 sugar plants, accounting for 40% of the sugar market in Poland. Here you can see one of the sugar plants located in the village Dobrzeje. I will add that the Institute of Biochemistry and Biophysics is a leader of these studies. Next slide. The studies concern production of fermentation gases during anaerobic digestion of biomass. Anaerobic digestion is a complex and multi-step conversion of biomass to methane and carbon dioxide, resulting from the metabolic activity of many groups of microorganisms. Anaerobic digestion involves 4 main stages: hydrolysis of polymeric organic matter to monomers. In our case, byproducts and waste of the sugar beet industry are used as biomass. The second step is acidogenesis. The third step is acetogenesis, and the fourth, methanogenesis. As you know, the final product of anaerobic digestion is biogas containing methane, which is used by man as an energy source. However, it should be emphasized that during acidogenesis, hydrogen-yielding processes occur. So we can conduct anaerobic digestion in 2-step systems to separate acidogenesis from acetogenesis and methanogenesis to produce biohydrogen and biomethane in separate bioreactors. Our efforts are focused on the acidogenic step of anaerobic digestion and selection of hydrogen-yielding processes. Why the selection is required? Because during acidogenesis, many other competitive types of fermentations can occur. Next slide, please. What we select? Here you can see 2 main types of hydrogen-yielding fermentations under mesophilic conditions. They are glycolytic fermentations, Clostridium type fermentation, and Enterobacteriaceae type fermentation. As you can see from the chemical reactions, the efficiency of hydrogen production during dark fermentation is low and equals 33%. But it's under condition that the only other fermentation product is acetate. Under the rare operating conditions, much less hydrogen is produced because many short fatty acids are formed. Next slide, please. Hydrogen can be also produced during the transformation of products of other fermentation types. The relevant hydrogen-yielding metabolic pathway is the conversion of lactate and acetate to butyrate, hydrogen, and carbon dioxide. The conversion results from symbiotic interactions between lactic acid bacteria and hydrogen-yielding bacteria, called cross-feeding of lactate, previously recognized for gastrointestinal bacteria. On the other hand, lactic acid bacteria are regarded as factors inhibiting hydrogen production due to competition for substrate. Therefore, our research focuses on the relationships between lactic acid bacteria and hydrogen-producing bacteria. Next slide, please. Here you can see a simplified scheme of our 2-stage anaerobic digestion system processing byproducts of the sugar beet industry. As a byproduct, we use sugar beet molasses containing 50% of sucrose. One bioreactor is for acidogenesis to produce hydrogen-rich fermentation gas, and the second bioreactor is for acetogenesis and methanogenesis to produce biogas. We use this system for basic research to study microbial communities and metabolic pathways of biohydrogen and biogas production. We also use this system for demonstration of an innovative technology for the continuous production of biohydrogen and biomethane on different scales. We recognize technical aspects and operating conditions of the processes. Next slide, please. Here you can see two-stage installations for biohydrogen and biogas production from sugar beet molasses at 3 different scales. Laboratory scale in our institute, it is technological readiness level 3. Tenfold in large installation, it is technological readiness level 4. And a quarter scale installation, it is technological readiness level 5. Both of them, it means tenfold in large and a quarter scale installations, are located and operate in Dobrzyn Sugar Factory. The studies were described in Bioresource Technology +1 and Polish Journal of Environmental Studies. Next slide, please. We use the hydrogen-yielding bioreactors from the installations to explore dark fermentation microbial communities and metabolic pathways of biohydrogen production. We selected bioreactors with medium and low efficiency of biohydrogen production and searched for differences in the microbial community's composition and metabolic activity. Next slide, please. We identified a relevant factor for hydrogen production. It's a community balance between dark fermentation bacteria and lactic acid bacteria. In the bioreactors with efficient hydrogen production, the ratio between dark fermentation bacteria and lactic acid bacteria was 2:1. In the bioreactors with a low hydrogen production, we observed 2 cases of solvatogenesis. In the first, a multiply predominance of lactic acid bacteria over hydrogen producers was observed, with lactate and ethanol as dominant fermentation products. In the second case, a multiply predominance of Clostridia over lactic acid bacteria with a high concentration of different short-chain fatty acids. Undoubtedly, stability of the hydrogen-producing microbial community is determined by technical and operating conditions. But in my opinion, a breakthrough in the research on hydrogen production by dark fermentation will be finding a mechanism or factor promoting metabolic shift from efficient hydrogen production to solvatogenesis, and finding on molecular level. And our results indicate that cross-feeding of lactate favors hydrogen production and is a relevant pathway in a high efficiently hydrogen producing microbial communities. And this study was published in Frontiers in Microbiology. Next slide, please. And we studied ability of dark fermentation microbial communities to the conversion of lactate to butyrate in batch cultures. The communities unable to this conversion, labeled in red in the slide, show a low richness, are composed mainly of lactic acid bacteria, produce mainly lactate and ethanol, and decrease pH. The communities able to convert lactate to butyrate, labeled in green in this slide, are composed of the genus Clostridium and lactic acid bacteria. The biodiversity of microbial communities increases with the ability to conversion. The pH is in the range 5 to 6. And these results were published in Microbiome. Next slide, please. We also identified Ascomycetes yeast as a new factor inhibiting hydrogen production. Yeasts compete for substrate, change fermentation type, cause metabolic shift, and pH decrease. Additionally, they produce killer toxins. All the factors disturb microbial community and cause dramatic drop in hydrogen production. And these results were published in International Journal of Hydrogen Energy. So summing up the acidogenic step, here I have one more slide, but you have to listen to only. So summing up the acidogenic step, the course of the microbial processes at the acidogenic stage of anaerobic digestion determines the efficiency of hydrogen production. There are many biological factors that disturb hydrogen production by dark fermentation, including competition for substrate and metabolic shift. Interestingly, regardless, and please one more slide, next slide. Interestingly, regardless of the predominant type of fermentation at the acidogenic step, methane production seems to proceed similarly. And these are conclusions from our studies, where lactate, acetate, propionate, and butyrate that are universal products of acidogenesis were used as a feedstock in artificial media lactate, acetate, propionate, and butyrate that are the universal products of acidogenesis. So we used as a feedstock artificial media containing one dominant product of acidogenesis, lactate, acetate, propionate, or butyrate. And the artificial media were processed by methane-yielding microbial communities in a long-term system in UASB bioreactors. Stable isotopic analysis of carbon and hydrogen evidenced that acetate and lactate favored the acetotrophic pathway, whereas propionate and butyrate favored the hydrogenotrophic pathway of methane formation, of course. Our hypothesis is that the substrate may first of all change the metabolic activity of the bacteria, methanogens, rather than the composition of the microbial community. This requires confirmation using metatranscriptomic analysis. And these results were published in Biotechnology for Biofuels. Next slide, please. Our studies on methanogenesis can help to understand and improve the operation of biogas plants and help to realize why technologies of biogas production are commercially available. Here you can see our implementation works. It was modernization of the wastewater treatment plant to use biogas for energy in the sugar factory. The wastewater treatment plant operates during the sugar campaign and processes spent beet transport water. It produces biogas containing up to 75% of methane. Until recently, the accumulated biogas was burnt and released into the atmosphere. Next slide, please. And now, after the modernization, the biogas is used as a fuel in a cogenerator system for energy production. The produced energy exceeds twice the energy demand of the wastewater treatment plant. Here, we can see wastewater treatment plant in Dobrzelin sugar factory after modernization. The new facilities are biogas purification system, biogas storage tank, and cogenerator. Our implementation reduces emission of greenhouse gases and increases contribution of renewable energy in the total energy pool. It's noteworthy to add that the daily balance of electricity produced reaches approximately 200% of the wastewater treatment plant demand. Next slide, please. All the activities are an excellent example of fruitful cooperation between a research unit and an industrial partner towards development of innovative technologies. Next slide, please. And here you can see new facilities on the wastewater treatment plant site from a bird's eye view. Number 1 and number 2, labeled in yellow, they are the facilities connected with modernization works to use biogas for energy. Number 1 is biogas tank and system for biogas purification, and number 2 shows localization of cogenerator. And number 3, labeled in red, it is two-stage installation producing biohydrogen and biogas from sugar beet molasses. Next slide, please. Summary. What will the audience should learn from the presentation? First issue, challenges and limitations of biohydrogen production during acidogenic step of anaerobic digestion include low efficiency of hydrogen production via dark fermentation. The theoretical maximum value is 33%. Metabolic shift and substrate competition between microorganisms, replacement of hydrogen-yielding fermentations by many other competitive types of fermentation, excretion of antibacterial factors by one group of microorganisms, inhibiting the growth of others, and necessity of the selection of hydrogen-yielding processes by specific operational conditions. And second issue, understanding the reasons for the immaturity of biohydrogen production technology compared to the advanced biogas production solutions. The reasons are biohydrogen is formed as an intermediate product of anaerobic digestion during acidogenesis, whereas biomethane is the final product of the anaerobic digestion process, regardless of what type of fermentation dominates the acidogenic, in the acidogenic stage. Limitations of biohydrogen production mentioned about. Third issue, I tried to show different perspectives for sugar factories. A sugar factory can be a place for the development of innovative technologies. And fourth issue, I presented, I hope, an excellent example of fruitful cooperation between a research unit and an industrial partner toward the new solutions, allowing for the diversification of the use of byproducts and waste in the sugar factory, demonstration of practical applications of scientific fundings, creation of the needs for qualified staff in rural areas. Next slide, please. Here you can see a list of the relevant publications. There are papers published in Biotechnology for Biofuels, Frontiers in Microbiology, Microbiome, Microbial Cell Factories, International Journal of Hydrogen Energy, Polish Journal of Environmental Studies, PLOS ONE, Bioresource Technology, and a chapter in a book. Next slide, please. And we cooperate not only with the sugar industry, but also with other scientific institutes and universities. Here you can see our institute, the Polish sugar industry company, as well as University of Arizona from USA, also University in Padua. We cooperated with Stefano Campanaro, and Polish institutions like University of Wrocław, Institute of Agriculture and Food Biotechnology, in Warsaw, Warsaw University of Life Sciences, University of Silesia, in Katowice, in Poland. And special thanks are due to Dr. Anna Detman from our institute, who is a main investigator in all the projects. And thank you for your attention.